Hello everyone. In today's session, we are going to learn about immunization, which is also called as vaccination. Immunization is defined as the process whereby a person is made immune or resistant to an infectious disease, typically by the administration of a vaccine. Over the last century, vaccination has been the most effective medical strategy to control infectious diseases. Smallpox has been eradicated worldwide, and polio has been almost eradicated. Most viral and bacterial diseases, especially those affecting children worldwide, are now preventable by vaccine. Vaccine is any preparation intended to produce immunity to a disease by stimulating the production of antibodies. Vaccine is actually an immunobiological substance designed to produce specific protection against a given disease. It stimulates the production of protective antibody and other immune mechanism. Vaccine provide active immunity to the body by stimulating the immune system, which produces antibodies against disease-producing organism. Vaccines may be prepared from live, modified organisms, inactivated or killed organisms, extracted cellular fractions, toxoids, or combination of these. Based on these, there are different types of vaccines like live vaccine, inactivated or killed vaccine, then subunit vaccines like toxoids, protein vaccines, recombinant protein vaccine, polysaccharide based vaccine, conjugated vaccine, combinations of different vaccines, and some future prospects, which we are going to see in detail in this session. Live vaccines are prepared from live attenuated organisms. These organisms have been passed repeatedly in the laboratory in tissue culture or chick embryos and have lost their capacity to induce full-blown disease but retain their immunogenicity. These live vaccines are more potent than killed vaccines because live organisms, they multiply in the host and result in antigenic dose which is larger than what is injected. Live vaccines have all major and minor antigenic components. They engage certain tissues of the body, as for example, intestinal mucosa by the oral polio vaccine. And there may be persistence of latent virus. But there are certain contraindications for giving live vaccines, like persons with immune deficiency disease, those who are having suppressed immune response because of malignancy, those who are on treatment with corticosteroids, alkylating agents, antimetabolic agents, or radiation. And pregnancy is another contraindication unless the risk of infection exceeds the risk of harm to the fetus of some live vaccines. When two live vaccines are required, they should be given simultaneously at different sites or with an interval of at least 3 weeks. Immunization is generally achieved with a single dose, except polio vaccine, which needs 3 or more doses to be given at spaced interval to produce effective immunity. Live vaccines produce durable immunity, but not always as long as that of natural infection. These vaccines must be properly stored to retain effectiveness. Some examples of live vaccines are like for bacterial diseases, BCG, that gives protection against tuberculosis, then typhoid oral. Viral vaccines like oral polio vaccine, yellow fever, measles, rubella, mumps, chickenpox, influenza, etc. And rickettsial diseases like epidemic typhus vaccine. Inactivated or killed vaccines. Here the organisms are killed by heat or chemicals. Inactivated vaccines are produced by growing virus or bacteria in culture media and then inactivating them with heat or chemicals. Usually the formalin is used. When injected into the body, they stimulate active immunity. They are usually safe but less efficacious than the live vaccines. Usually, it requires a primary series of 2 or 3 doses to produce adequate antibody response, and mostly booster is required. Duration of immunity varies from months to many years. Inactivated polio vaccine has been quite an effective vaccine, the widespread use of which in certain countries has led to the elimination of the disease. They are generally administered by subcutaneous or intramuscular route. The only absolute contraindication to the inactivated or killed vaccine is severe local or general reaction to the previous dose. Because the vaccine is inactivated, the ineffective infective agent cannot grow in the vaccinated individual and therefore cannot cause the disease even in an immunodeficient person. Some examples of inactivated or killed vaccine are typhoid, cholera, pertussis, plague, then viral diseases like rabies, in injectable polio vaccine, influenza, hepatitis B and A, Japanese encephalitis, and KFT. Then a vaccine can also be made of single or multiple antigenic components of a microorganism, which can stimulate a specific immune response sufficient to produce from the relevant pathogen infection or from the clinical manifestation of the disease. Depending on the molecular composition of the purified antigen used to prepare the vaccine and on the techniques applied to obtain the final material used as a vaccine, different types of subunit vaccines can be defined. Like toxoids, which is the most commonly used subunit vaccine. Here, the toxins produced by certain organisms are detoxicated and used in the preparation of vaccine. Antibodies produced, they neutralize the toxin produced during infection rather than acting upon microorganism. They are highly efficacious and safe. Diphtheria and tetanus vaccines are examples of these toxoids. In case immunization with a single protein or combination of proteins from a pathogen is sufficient to produce a protective immune response against that particular microorganism. The approach of a protein-based vaccine is appropriate, and these proteins can be purified from in vitro cultures of a pathogenic microorganism. A cellular pertussis vaccine, it is licensed vaccine currently available, which contains contain from 2 to 4 different proteins purified from Bordetella pertussis and are able to confer protection against whooping cough comparable to that obtained with a whole cell vaccine. Another example of this protein vaccine is influenza vaccine composed of hemagglutinin and neuraminidase purified form the inactivated influenza virus. A drawback of a clean vaccine preparation containing pure recombinant protein is the reduced immunogenicity that may require the addition of an adjuvant to achieve enhanced efficacy. As, for example, in the hepatitis B vaccine is made by inserting a segment of hepatitis B virus gene into a yeast cell. The modified yeast cell produces large amount of hepatitis B surface antigen, which is purified and harvested and used to produce the vaccine. The recombinant hepatitis B vaccine is identical to the natural hepatitis B surface antigen, but it does not contain virus DNA and it is unable to produce infection. This technique is called as recombinant protein vaccine. Recombinant DNA technology is used for the heterologous expression of protective protein antigen. The advantages of this is it avoids the problem related to growing and manipulating large amount of pathogen from which the antigen is purified. Recombinant proteins are generally better purified from cultured microorganisms, resulting in cleaner vaccine preparation with a better safety profile. The drawback of this recombinant protein vaccine is it requires the addition of an adjuvant to achieve enhanced efficacy. Then the surface of many pathogenic bacteria are covered by a capsular cell that is made up of polymeric glycans. This polysaccharide coat entirely covers the bacteria and prevents it from the target of host immune response. But still, there are certain mechanisms like complement-mediated killing, and opsonophagocytosis by which the antibodies to these surface polysaccharides can clear the bacteria. Hence, stimulation of an antibody response against the surface polysaccharide of pathogenic bacteria is a strategy for the development of vaccine against capsulated bacteria. The drawback of polysaccharide-based vaccine is the immune response they elicit are often serotype specific because chemical structure of capsular polysaccharides varies not only between bacteria of different species but also between different strains of same bacteria. Next is conjugated vaccine. The polysaccharide antigens are chemically linked or conjugated to a protein that T cells recognize. Then this conjugate vaccine can elicit strong immune response and immune memory in young children. Children under 2 years of age, they do not respond well to antigens such as polysaccharide, which produces antibodies via T cell dependent mechanism. The drawback of conjugated vaccines are they are also serotype specific and therefore multivalent formulations are required to achieve protection against multiple serotypes. For example, pneumococcal and meningococcal vaccines. If more than one kind of immunizing agent is included in the vaccine, it is called as mixed or combined vaccine. The aim is to simplify administration, reduce costs, and minimize number of contacts of patient with health system, reducing the storage cost, improving the timelines of vaccination, and facilitating the addition of new vaccine into the immunization program. One more advantage is that it does not increase the risk of reactions and can in fact lead to an overall reduction in adverse reactions. Pentavalent vaccine, DT, MMR, these are some examples of combined vaccines. Polyvalent vaccines are prepared from 2 or more strains of same species, like poliovirus, which is prepared from 2 or 3 strains of the poliovirus, then influenza virus. Autogenous vaccines are the vaccines where the organism in the vaccine is obtained from the same patient. So these are different types of vaccines. Apart from this, there are some other components in the vaccine, like adjuvant. These are the substances that are added to the vaccine with an intent of potentiating the immune response. The advantage is that the greater amount of antibody is produced and lesser quantity of antigen and fewer doses are required. Examples are aluminum phosphate and aluminum hydroxide, etc., which are added in the DPT vaccine. The mechanism is that they form a local granuloma which retains the antigen and releases it slowly. Then antibiotics, which are used during the manufacturing phase to prevent bacterial contamination of the tissue culture cells in which the viruses are grown. For example, MMR vaccine and the IPV each contains less than 25 micrograms of neomycin per dose. Persons who are known to allergic to neomycin should be closely observed after vaccination so that any allergic reaction can be treated at once. Then there are certain preservatives. These are the chemicals which are added to killed or subunit vaccine in order to inactivate viruses, detoxify bacterial toxins, and to prevent serious secondary infections as a result of bacterial or fungal contamination. Then there are stabilizers. These are the compounds added to confirm product quality or stability of vaccines. For a variety of manufacture related issues like controlling the pH, freeze drying, and preventing antigens from adhering to the sides of glass vials with a resultant loss in immunogenicity. The examples of these stabilizers are potassium or sodium salts, lactose, human serum albumin, and a variety of animal proteins such as gelatin and bovine serum albumin. Apart from this, the molecular biology and modern technologies are combining to devise novel approaches to vaccine development. Few of these examples we are enlisting here, but the ultimate success of these new approaches remain to be determined. The first one is the use of recombinant DNA techniques to insert the gene coding for the protein of interest into the genome of an avirulent virus that can be administered as the vaccine. A recent example of this kind of vaccine is coronavirus vaccine, which is a recombinant replication deficient chimpanzee's adenovirus vector encoding the SARS-CoV-2 spike glycoprotein produced in genetically modified human embryonic kidney. Then including in the vaccine only those subviral components needed to stimulate protective antibody, thus minimizing the occurrence of adverse reactions to the vaccine. Use of purified proteins isolated from purified virus or synthesized from cloned genes. Expression of cloned genes sometimes result in formation of an empty virus-like particles. Use of synthetic peptides rather than the entire viral protein. This avoids the possibility of reversion to virulence since no viral nucleic acid would be present, although the immune response induced will be weaker than the induced by intact protein. Development of edible vaccines, whereby the transgenic plants synthesizing antigens from pathogenic viruses may provide new cost-effective ways of delivering vaccine. Use of naked DNA vaccines, potentially simple, cheap, and safe, in which recombinant plasmids carrying the gene for protein of interest are injected into hosts and the DNA produces the immunizing protein. Now, these are the different types of vaccines that we have seen. Now, we'll see the universal immunization program, which is implemented in our country. Before that, some key achievements under this universal immunization program. The program was started in 1978 and a lot of modifications have been made from time to time. Now we'll see the national immunization schedule for infants, children, and pregnant women. Start with the pregnant woman where they are given 2 doses of TT first as early as possible in the pregnancy and second dose 4 weeks after the first dose. If the woman has received 2 doses in a pregnancy within the last 3 years, only a booster is required. Second dose of TT or booster dose should be given before 36 weeks of pregnancy. However, this can be given if more than 36 weeks have passed. Give TT to a woman in labor if she has not previously received any TT injection. Schedule for the infants. At birth, BCG, hepatitis B, birth dose, and OPV 0 dose is given. Then at 6, 10, and 14 weeks, OPV 1, 2, 3, and pentavalent vaccine, which contains diphtheria, pertussis, tetanus, hepatitis B, and H influenza B. So it gives protections against 5 diseases and it is called as pentavalent vaccine. So 3 doses of this pentavalent vaccine is given at 1, 2, and at 6, 10, and 14 weeks. Then inactivated polio vaccine is given at 6 and 14 weeks. Similarly, rotavirus vaccine, which is now introduced in phase-wise manner. So wherever applicable, it is given at 6, 10, and 14 weeks. Again, pneumococcal conjugate vaccine can be given at 6 and 14 weeks, and at 9 completed months, a booster is given. Measles, or nowadays it is the MR vaccine, which is introduced in phases, replacing the measles vaccine in the UIP schedule. So it is given at 9 completed months. And if it is delayed beyond 12 months, ensure that minimum 1 month gap is there between 2 MR doses. Japanese encephalitis vaccine also has been introduced in selected endemic districts. And if the first dose, it is given at again at 9 to 12 months. If it is delayed beyond 12 months, ensure that there is minimum 3 months gap between 2 Japanese encephalitis doses. Vitamin A first dose is also given along with the measles at 9 months of age, which is of 1 lakh international unit. Then for the children more than 1 year of age, DPT booster, MR second dose, OPV booster, Japanese encephalitis, they're all given between the age of 16 to 24 months. Vitamin A, the second dose onwards, the second to ninth dose of vitamin A can be administered to children between 1 to 5 years of age during biannual rounds in collaboration with ICDS. Then DPT booster, second dose is given at the age of 5 to 6 years and TT is given at 10 years and 16 years. Apart from these vaccines, the human papillomavirus vaccine is presently not in the schedule, and TD, that is tetanus diphtheria, will replace the TT vaccine, which will be added in the schedule. This was about the immunization and the vaccine. Now, another important topic that we are going to discuss with regard to vaccine is the cold chain. Cold chain is a system of storage and transport of vaccine at low temperature from the manufacturer to the actual vaccination site. The cold chain system is necessary because vaccine failure may occur due to failure to store and transport under strict temperature controls. In other words, the success of national immunization program is highly dependent on supply chain system for delivery of vaccines and equipments with a functional system that meets 6 rights of supply chain. That is, the right vaccine in the right quantity, at the right place, at the right time, in the right condition. There is no temperature breaks in the cold chain and at the right cost. So these 6 rights are there. So this is how the vaccine will be transported from the manufacturer to the recipient by various routes and stored at various places. At all these places, the strict temperature has to be maintained. The key elements of cold chain system are the persons who manage the vaccine storage and distribution, the vaccine and cold chain handler at each cold chain point, then equipments to store and transport the vaccine and monitor temperature, and the procedures to ensure correct utilization of equipment and ensure vaccines are stored and transported safely. It is the responsibility of healthcare workers to manage the cold chain at the session site and sometimes at the cold chain point also. The temperature requirements for the vaccines. Some vaccines are sensitive to freezing and some to heat and others to light. The potency of the vaccine, that is, the ability to adequately protect the vaccinated patient, which will diminish when the vaccine is exposed to inappropriate temperatures. Once this potency is lost, it cannot be regained. And to maintain the quality, the vaccine must be protected from temperature extremes. Here is the list of vaccines which are sensitive to heat, light, and freezing. The OPV vaccine is sensitive to heat, while the measles and MR vaccine, they are sensitive to heat as well as light. The BCG, rotavirus vaccine, and Japanese encephalitis vaccine, they are relatively heat stable, but they are sensitive to light. Those vaccines which are sensitive to light are always stored in amber-colored bottle. OPV, measles, and OPV, measles, MR, and BCG, rotavirus, and Japanese, they are not damaged by the freezing. While the hepatitis B, pentavalent vaccine, pneumococcal conjugate vaccine, then IPV, DPT, and DT, they are relatively heat stable, but they they are sensitive to freezing and they freezes at minus 0.5 degrees Celsius, and that thereby it should not be allowed to freeze. At PHC level, all vaccines are kept in the ILR for a period of 1 month at temperature of +2 to +8 degrees Celsius. So the vaccine which is most sensitive to heat is the BCG vaccine after reconstitution. And this is the sequence in which the sensitivity goes on decreasing, while the pentavalent hepatitis B and PCV vaccine are the least sensitive to heat. But the hepatitis B vaccine is most sensitive to freezing, followed by PCV, pentavalent vaccine, and then IPV, DPT, and TT are the least sensitive to freezing. So we will have an overview of these cold chain equipments. The cold chain equipments, there are four categories. First, required for the storage, then transportation, then there are associated equipments and temperature monitoring devices. For the storage, electrical, solar, and non-electrical equipments are used. Electrical equipments are walk-in cooler, walk-in freezer, ice line refrigerator, and deep freezer. Then solar refrigerators are used, while the non-electrical storage equipments are cold box and vaccine carrier. For the transportation, refrigerated van, insulated van, cold box, vaccine carrier, and ice packs are used. Along with that, we require stabilizer, generator, and inverter to maintain the temperature. And to monitor the temperature, we have thermometer, electronic data logger, freeze indicator, real-time temperature monitoring device. We'll see some important equipments out of this. Walk-in freezers, these are prefabricated modular polyurethane foam insulated panel which assembles cold room with two identical refrigeration units and a standby generator set to provide the uninterrupted water power supply. They maintain the temperature between minus 15 degrees Celsius to minus 25 degrees Celsius. They are installed at national, state, and regional vaccine store. They are used for bulk storage of OPV vaccine during pulse polio vaccination campaign and for preparation of frozen ice packs for vaccine transportation. Similarly, there are walk-in coolers which are also made up of a prefabricated modular polyurethane insulated panel assembled cold rooms. They maintain the temperature of plus 2 degrees Celsius to plus 8 degrees Celsius. They are used for storage of large quantities of all the vaccines. They have two identical cooling units and also a standby generator. They are installed at government medical store depot, state and regional vaccine stores. Walk-in coolers and walk-in freezers, they come with continuous temperature record and alarm system. Then there are deep freezer, which operates on a vapor compression system similar to any conventional type of refrigerator, which operates at 220 volts AC main supply. Only thing is that it is having top opening lid, which prevents the loss of cold air during the door opening. Temperature is maintained between minus 15 to minus 25 degrees Celsius, similar to walk-in freezers. They are used for storing the OPV at the district level and above only, and also for freezing of ice packs at sub-district level only. Deep freezer has less holdover time as compared to ice-lined refrigerator, which depends on number of frozen ice packs in it and the frequency of opening the lid. We will see what this holdover time is later. The deep freezers, which is used for storing vaccines, should not be used for preparation of ice packs. It may increase the cabinet temperature and can be potentially harmful to the vaccines, especially the OPV. However, adequate frozen ice packs can be kept permanently inside the vaccine storing deep freezer for increasing the holdover time. There are some do's and don'ts with the deep freezers. Do's: use deep freezer only for the preparation of ice pack at sub-district level cold chain points, that is primary health center, community health center, and sub-center level. Use deep freezer to store OPV at district level. Keep frozen ice packs in the vaccine during deep freezer to increase the holdover time. Then don'ts: do not keep any vaccine in the deep freezer at sub-district level. Never keep diluents in the deep freezer. At district level, do not use the same deep freezer for simultaneously storing vaccine and preparing ice packs. This is how the ice packs should be arranged in the deep freezer. The blue colored ones are the unfrozen ice packs, while the gray colored are the frozen ice packs. So there are two compartments, large compartment and the small compartment. In the small compartment, the frozen ice packs are stored and arranged horizontally in layers, similar to cold boxes. While this blue color, they are the unfrozen ice packs, while the frozen ice packs, they are stored only up to the half of the height of the large compartment. This is the brick-layered ice packs in deep freezers. They should not be stored. They should always be stored like this in a brick-layered pattern with a space in between for air circulation. Iceland refrigerators, they also operate on the same system as the domestic refrigerator. They maintain the temperature between plus 2 degrees Celsius to plus 8 degrees Celsius. They are used to store vaccines at district and sub-district level. It has having a top opening lid to prevent the loss of cold air during door opening. It can keep the vaccine safe with a minimum 8 hours of continuous electricity supply in a 24-hour period. They are available in two sizes. Large ones are available for the district headquarters, while the small ones are available for primary health centers. This is how the vaccines should be stored in the ice-lined refrigerator. At the top, there should be diluent, followed by the vaccines which are sensitive to heat, freezing, that is hepatitis B, followed by a pentavalent vaccine, and the vaccines which are sensitive to heat should be stored at the bottom, that is OPV, measles, BCG, Japanese encephalitis, rotavirus, etc. While in the other section, the written vials are stored in separate container. The do's and don'ts for the ice-lined refrigerators. Iceland refrigerators keep all the vaccines, including those returned under the open vial policy, in the basket supplied along with the Iceland refrigerators. Diluents should be stored at plus 2 to plus 8 degrees Celsius at least 24 hours before use. Leave space in between the vaccine boxes. Place a thermometer in the basket in between the vaccines. Keep freeze-sensitive vaccines at the top of the basket. Keep heat-sensitive vaccines at the bottom of the basket, and arrange the vaccines as per their expiry dates. Early expiry should be above the further expiry ones. Do not store any other drugs or non-UIP vaccines in the ILR. Do not use ILR. Do not open the ILR frequently. Do not keep the food or drinking water in the ILR. Do not keep vaccines which have expired and have crossed the discard point of vaccine vial monitor. We will see this later. Do not disturb the thermostat setting unless needed. Do not place heavy weight on ILR. Do not store excess stock of vaccine, that is more than the maximum stock. And do not store any reconstituted vaccine vial. Now, what is the hold or time of the equipment? It is the time taken by the equipment to raise the inside cabinet temperature from its temperature at the time of power cut to maximum temperature limit of its recommended range. So when the power is there, the temperature will be maintained between either plus 2 to plus 8 degrees Celsius for the ice-lined refrigerators or walk-in coolers, and minus 15 to minus 25 degrees Celsius for deep freezers or walk-in freezers. When the power is cut off, how much time it requires to cross this temperature limit is called as hold-over time. Hold-over time depends on certain factors like ambient temperature. More the ambient temperature, less will be the hold-over time. Frequency of opening the lid and use of basket. Quantity of vaccines kept inside with adequate space between the containers, whether the containers are empty or loaded, and the condition of ice packs, like whether they are frozen, partially frozen, or melted. So these factors affect the hold-over time of the equipment. In domestic refrigerators, they can also be used to store the vaccine, which maintain the temperature between plus 2 to plus 8 degrees Celsius with a hold-over time of 4 hours. They can be used for storage of vaccine at private clinics and nursing homes, provided there is continuous water supply, power supply. Then there are cold boxes which are supplied to all peripheral centers. This is non-electrical equipment which is mainly used for transportation of vaccines. Before the vaccines are placed in the cold boxes, fully frozen ice packs are placed at the bottom and sides. Vaccines are first kept in cartons or polythene bags. They should not be kept in direct contact with the ice packs. While some DPT, DT, and TT vaccines and diluents, they should not be kept in direct contact with the frozen ice packs. So this is the view of cold box where it is first lined from all sides with ice packs, and then the vaccines are kept inside this. Then there are vaccine carriers which are used to carry small quantities of vaccines like 16 to 20 vials for outreach sessions. 4 fully frozen ice packs are used for lining the sides. Again, vials of DPT, DT, and TT and diluents, they should not be kept in direct contact with the frozen ice packs. The lid should be tightly closed. So this is the vaccine carrier which most of you must have seen. Do's and don'ts for the vaccine carrier. Do's: place vaccines and diluents in cartons or polythene bags to ensure labels are protected. Use well-sealed conditioned ice packs in the vaccine carrier. Ensure collection of vaccines in the vaccine carrier on the session day itself. Close the lid tightly and securely, and keep the interior of the vaccine carrier clean and dry after every use. Don'ts: never use day carriers which contains 2 ice packs or thermos flasks for routine immunization. Never use any screwdriver or any other sharp shaft to open the lid of vaccine carrier. Do not drop, knock, or sit on the vaccine carrier. Do not leave the vaccine carrier in the sunlight, and do not leave the lid open once packed. Day carriers are used to carry small quantities of vaccine, that is 6 to 8 vials, to a nearby session. Only 2 fully frozen ice packs are used for the lining and used only for few hours period. This is same as vaccine carrier but small in quantity. Ice packs, they contain water and no salt should be added. They should be filled up to the level marked on the side, and if there is any leakage, it should be discarded. So these are the ice packs, and there is a lining on the ice packs up to which the water should be filled. Do's and don'ts for the ice packs: fill the water only up to the level marked on the side. Here it is. To leave 10 mm room for expansion of water after it freezes. While filling, keep the ice pack vertically upwards under the tap so that it will overflow after reaching the desired level. Fit the stopper and screw on the cap tight. Check and ensure that ice pack does not leak. Clean the outer surface of ice packs with dry cloth before putting into the deep freezer. Keep ice packs horizontally, not flat, in a crisscross manner, that is in a brick-laid manner in deep freezer. Keep gap or breathing space between ice packs for freezing to be faster and uniform. Ensure that ice packs are frozen rock solid. Don'ts for ice packs: do not use the ice packs that are cracked and are without cap or cork. Do not use ice packs with leakage. Discard those ice packs. Never add salt to the water as it lowers the temperature to sub-zero level, which is not recommended. And do not fill ice pack every time before use. Same water can be used repeatedly. So this is a summary of all the equipments which are used. Deep freezer, the temperature which is required, and the capacity and the hold out time. As you can see, the hold out time is more for the ice-line refrigerators. It is more even for the cold boxes. The temperature required for the deep freezer is -15 to -25 degrees Celsius. For the ILR, it is +2 to +8 degrees Celsius. For all non-electrical equipments, the temperature is +2 to +8 degrees Celsius. The hold out time for the deep freezer, large ILR, and deep freezer small size are 2 hours, 30 minutes for the deep freezer, and 20 hours for the ILR. For cold boxes, it is 96 hours. For small size cold boxes, it is 48 hours, and for vaccine carriers, it can be stored up to 36 hours. 43 degrees Celsius is the ambient or the surrounding temperature which is there. What is vaccine vial monitor? These are the temperature monitoring devices that routinely accompany vaccines throughout the entire supply chain. Vaccine vial monitor is a chemical indicator label attached to the vaccine container by the manufacturer. It records cumulative heat exposure through a gradual change in color. The purpose of vaccine vial monitor is to ensure that heat damaged vaccines are not administered, to decide which vaccine can safely be kept after a cold chain break occurs, thus minimizing the unnecessary vaccine wastage. And how to read this vaccine vial monitor? The vaccine vial monitor start color of the inner square is never pure white. It is always slightly purple and begins lighter in color than the outer circle. From this point forward, the inner square starts to darken in the color until the temperature or the duration of heat reaches a level which degrades the vaccine, at which time the vaccine vial monitor's discard point will be evident. So you can use the vaccine till the color of inner square is lighter than the outer circle. When the color of inner square becomes same as that of outer circle or it becomes more darker, then the vaccine should not be used. This vaccine vial monitor ensures the heat damage of the vaccine. While the vaccines which are sensitive to freezing, how to check for this freeze damage to the vaccine? Freeze sensitive vaccines, they will get spoiled if they get frozen. The causes may be improper storage in the ice-lined refrigerator, cold climates, or if the ambient temperature itself is less than 0 degrees Celsius, storage and transport with non-conditioned frozen ice packs, and defective ILR, incorrect thermostat adjustment, untrained or improperly trained staff. These may be the reasons why the vaccine may be exposed to the freezing and it gets damaged. The shake test is used to check whether freeze-sensitive vaccines have been damaged by exposure to temperature below 0 degrees Celsius. After it has thawed, a vial of vaccine that has been frozen no longer than the appearance of a cloudy liquid, but tends to form flakes that settle down at the bottom of the vial. So in the shake test, you have to take a vaccine vial which you suspect that may have been frozen. This is called as test vial. And take another vial which is called as control vial. This vaccine vial of the same antigen, same manufacturer, and same batch number as the suspect vaccine vial you want to test. Freeze solid this vaccine at minus 20 degrees Celsius overnight in the deep freezer. And this is the control vial and label accordingly to avoid its uses. Let it thaw. Do not heat it. Hold the control and the test vial together between thumb and the forefinger and vigorously shake the vials for 10 to 15 seconds. Place both the vials to rest on a flat surface side by side and observe them for 30 minutes. Compare the rate of sedimentation. If the sedimentation rate in test vial is slower than the frozen vial, the vaccine has not been damaged. It has passed the shake test and you can use the vaccine. It is not damaged. If the sedimentation rate is similar in both the vials, and if the sedimentation is faster in the test vial than in the frozen vial, then the vaccine is damaged. It is failed the shake test and do not use, and it should be notified to the supervisor. So this is where you can see the sedimentation rate, and this is the failed shake test. So this was about the maintaining the cold chain for maintaining the potency of the vaccine. Thank you.